Hello everyone. My name is Pranjal Tripathi, and today I'm going to present my presentation on the topic of uh, a concept in environmental genomics and introduction to the intersection of genomics and the environment. So, in this PPT, we are going to overview some key points and key topics of environmental genomics. First, that is introduction to the environmental genomics. Second is the key concept in environmental genomics. Third is technological advancement in environmental genomics, and the last that is the application in environmental genomics. So let's start this presentation by through. Uh, let's start this presentation introduction to the environmental genomics. What is environmental genomics? The environmental genomic is the study of genetic material of microbes directly obtained from the organism, from the environmental samples like water, soil, and air and sediment without isolating the organism first, analyzing how environmental factors influence gene expression and adaptation to the organism. The purpose of studying this topic is to explore diversity, ecosystem, environmental changes at the molecular level. The key concept in the environmental genomics. So, the key concept in the environmental genomics is to study how a genetic makeup of organism directly obtained from their natural environment using genomic tool to understand how organism adapt and responds to environmental changes at the molecular level. The field of environmental genetics and environmental genomics has enormous potential to affect our ability to accurately assess the risk of developing disease, identify and understand the basic pathogenic mechanism. So the key concept in environmental genomics, first that is diversity, genetical diversity. As we all know that on the on this planet Earth, we have a lot of genetical diversity within a species. So the genetic diversity talk about the biological variation in the DNA that occur within a species. It is important to allow species to adapt to a changing environment, which is especially important to face a rapid environmental changes. Genomic technology has made it possible to quickly and cheaply sequence entire genome, which has expanded our understanding of wild populations. Okay. For example, researcher can look on the genetic trait and influence the population diversity, disease resistant, etc. Metagenomics. So metagenomics and environmental genomics are the same thing only. The second name of environmental genomic is metagenomic. Is the study of collective genetical material uh, directly extract from the microbial community within a environmental sample, and researcher to analyze the genetic diversity and the functional potential of all microorganisms present in the habitat without needing to isolate the individual species in the lab. Functional genomic. Functional genomic is the very interesting thing uh, in environmental genomic. It is the unlike it is the it is not a traditional environmental study that might only look on the phenotypical changes, but it is allow researcher to examine which gene are being actively transcribed or responsible for the environmental stress, allowing researcher to understand the molecular mechanism behind adaptation and the survival in different environment. So as we all know that all microorganisms are different from each other. They have a specific functional gene which make them special, which make the organism to adapt in that environment and survive in that environment. So functional genomics is that only to study that functional gene. Technological advancement in environmental genomics. First, that is next generation sequencing, NGS, is a DNA sequencing technology that determines the order of nucleotide in DNA or RNA by sequencing multiple DNA fragment in parallel. NGS is the high throughput technology that had made it faster and less expensive to sequence the entire genome. So in NGS, we are just going to take a small sequence of DNA or RNA of organism and make a multiple fragments and we can and make multiple fragments from that small DNA or RNA sample, and we can easily or cheaply or fastly we can study the whole genomic of that organism. Second is bioinformatives. Bioinformatives is the environmental genomic, referred to the application of computation tools and algorithm to analyze large-scale genetic data from the environmental microorganism, allowing researcher to understand the microbial community. So basically, we that whatever we did in NGS, we took all the sequence of the individual microorganism or the sequence of gene of individual microorganism. So through bioinformative we can put that whole genomic of that microorganism using computation through an algorithm, and it is make easy for researcher to study that microbial community. So here is the dramatical explanation of uh, NGS and bioinformatives. The next point, that is application of environmental genomics. In this, we are going to talk about microbial ecology, bioremediation, climate change, and research, evolutionary studies. So, microbial ecology, understanding microbes community in the ocean, sea, and other environment. Bioremediation, uh, sorry, okay. Microorganism can be genetically modified to break down pollutant in soil and groundwater. This process is called bioremediation. Climate change research, understanding how microbial community responds to a climate change by analyzing their genetic material and functional potential under different microbial conditions. Evolutionary studies, studying the genetic evolution of microbial community within a environment, providing insight into the adaptation and mechanism and evolution. So that's it for this PPT, and I'm going to end this PPT. Thank you so much for listening me, and hope you understand the concept of environmental genomics.